Hey there, focused learners, and welcome to this explainer. I am incredibly excited to be hanging out with you today because we're going to be diving into an absolutely brilliant new computational neuroscience model out of MIT. And, spoiler alert, it essentially solves a massive mystery about human memory. You know, if you've ever wondered how your brain manages to store a literal lifetime of experiences without running out of space, or why our super advanced AI systems still struggle with basic memory compared to biology, well, you are in the exact right place. People often say the human brain holds somewhere around 2.5 million gigabytes of digital memory. But the real magic, it's not just that sheer mind-boggling volume. It's how the brain stores all that data without the whole system just collapsing. So let's dive right into this fascinating architecture of the mind.
Think about how your memory actually plays out in your day-to-day life. The researchers hit the nail on the head in our source paper when they say we can revisit scenes and events from only a few cues, as with Proust's famous Madeleine. I mean, think about it. Just the smell of a certain perfume, or the taste of a specific pastry, or, that highly specific, terrible smell of your middle school cafeteria. And suddenly, boom, you instantly reconstruct this incredibly vivid scene from your childhood. We call this episodic memory. It's rich, it plays out in a sequence, and it is entirely associative. We aren't searching our brains like a computer looking for a specific file name, right? Instead, we just pull on this one tiny thread of sensory input, and the entire beautifully complex fabric of that memory comes rushing back.
But this incredible everyday thing we all do brings us to a seriously massive biological mystery. So for decades, neuroscientists have known that the hippocampus is the memory center of the brain. If it gets damaged, we lose our episodic memory, those personal diaries that essentially make us who we are. But here's the kicker. That exact same brain region, using the exact same neural hardware, is also responsible for our spatial navigation. It's literally our internal GPS, mapping out our physical environments using things called place cells and grid cells. So here's the million-dollar question. Why on earth do these two completely different things, navigating the physical layout of your living room and remembering an abstract conversation you had 10 years ago, share the exact same address in your brain? To really get why biology built things this way, we kind of have to look at where our best artificial intelligence completely faceplants. In computer science, specifically with these content-addressable memory models known as Hopfield networks, there is this terrifying mathematical problem known as the memory cliff. And let me tell you, it is literally a developer's worst nightmare. It's this catastrophic failure where a neural network works absolutely perfectly, flawlessly, up to a very strict mathematical limit. But the exact second you add just one single memory past its capacity, it doesn't just politely reject the new memory. Nope, it perfectly and completely erases all of the previously stored memories. The whole shebang, everything, just vanishes into thin air. Just take a look at the graph we have up right now to clearly visualize that catastrophic failure. See that solid blue line? That represents your traditional Hopfield network. Along the bottom, we've got the number of patterns memorized, and going up the side is the amount of information the system actually retained. Notice how the blue line stays perfectly flat, holding on to perfect, pristine memory as new stuff is added until, boom, it hits a rigid wall and literally drops straight down to zero. That sheer, sudden drop-off right there, that is the memory cliff. The system is incredibly brittle. You add one piece of data too many, and the whole thing crashes. Luckily for us, that is absolutely not how human brains operate.
It's a pretty wild contrast when you put them side by side, and it really highlights the sheer magic of human biology. On one side, we have traditional Hopfield networks suffering from total catastrophic erasure the second they get overloaded. And on the exact opposite side, we've got us. Think about it. When you memorize a bunch of new phone numbers or try to learn a whole new language, you don't wake up the next day with zero memory of your own name or where you went to high school. Our biology pulls off this incredibly graceful trade-off between the pure capacity of what we can store and the precise detail of what we recall. You might forget exactly what color shirt you wore to the party, but you certainly don't forget the party completely. But the underlying mechanics of how the brain pulls this off and avoids that cliff, that has been a total mystery. Until now.
All right, which brings us to Section 1, the vector hash scaffold, a biological breakthrough. This brand new model from MIT basically proposes this brilliant mechanism for how the mind weaves spatial and episodic memory together. Okay, so vector hash, it actually stands for vector hippocampal scaffolded heteroassociative memory. I know, it's a massive mouthful of a name, but the logic underneath it is actually quite beautiful. To wrap our heads around it, let's just trace the biological flow of a memory. First step, raw sensory inputs. Everything you're seeing, hearing, tasting, they get processed in the cortex. Second, all that rich sensory info flows right into the hippocampus. And third, and this is absolutely the most crucial step of the whole model, the hippocampus starts talking back and forth with this highly specific group of neurons called entorhinal grid cells. These grid cells, they are the absolute key to the entire system working without falling off that cliff.
To really visualize what these grid cells are doing mathematically, check out these donut shapes, or mathematical toruses. When it comes to physical spatial navigation, these modules act sort of like an internal compass. They help you track exactly where you're standing in a room. But here is the extraordinary thing the MIT researchers realized. Even when you're dealing with non-spatial memory, like trying to remember that complicated conversation from 10 years ago, these grid cells are acting as low-dimensional mathematical modules. Because they literally cycle back on themselves over and over like a donut, they generate this massive repeating library of extremely robust, incredibly stable fixed points. And that happens whether you're mapping out the coordinates of a coffee shop or mapping out highly abstract conceptual memories.
All right, here is the absolute secret sauce of the whole thing. The foundational secret to biological memory is entirely separating the dynamics of error correction from the actual memory content itself. Just let that sink in for a second. In standard AI, the system uses the messy, noisy sensory details to try and build its own internal structure. And because the data is noisy, the structure itself becomes fragile, which is exactly why the whole thing eventually hits that cliff and crashes. But the brain is way, way smarter. It uses those grid cells we just talked about to create a pre-structured, rigid mathematical network, a scaffold. And it keeps that scaffold completely 100% separate from the messy sensory details we're actually trying to remember.
And to make all that crazy math a lot easier to visualize, let's head into Section 2, the sturdy clothesline, the brain's anchor points. This is such a fantastic metaphor. So instead of some chaotic shifting energy landscape that radically changes shape every single time you learn something new, the brain's grid cell scaffold actually acts like the orange structure you see here. It works like a pre-built, incredibly sturdy clothesline. The grid network provides these equally deep, perfectly spaced anchor points that simply do not move. So the arbitrary sensory content you experience every day, like seeing a rare bird or your best friend's face, none of that changes the underlying structure of the clothesline. The sensory data is literally just hooked onto these super stable points, hanging there nice and securely. Now you might be wondering, how does biology actually build this sturdy clothesline in the first place? Well, it happens through this fascinating process the researchers call strong generalization. Essentially, the neural weights flowing from the grid cells to the hippocampus, they're mostly random and fixed right from the start, but the weights going backward from the hippocampus to the grid are learned associatively. And here is the true magic of strong generalization. This setup mathematically generates an exponentially massive number of stable hooks on that clothesline. But, and this is huge, it only requires the brain to actually physically visit a tiny, microscopic fraction of those possible states to lock the whole network into place. Meaning, just a little bit of a baby physically crawling around their living room can build a mathematical memory scaffold robust enough to last them their entire life. I mean, what a masterclass in biological efficiency. Okay, so we've got our sturdy clothesline fully built. But what happens when we really start stress testing biology? Let's get into Section 3, graceful trade-off, testing biology limits. Check out the images of the bird here. This is explicit visual proof of what happens when the vector hash model is deliberately pushed past its limits. As the system is stretched past its optimal capacity, the memory doesn't just poof out of existence like it does in an AI memory cliff. Instead, the grid cells in the hippocampus still hit their fixed structural marks perfectly on the clothesline. But the sensory reconstruction attached to those marks, it just gets slightly fuzzy. You can see the recovered image of the bird slowly degrading into a blur. The system brilliantly trades high-fidelity sensory detail for overall storage capacity, making absolutely sure that you always get something useful back when you try to remember, instead of just a blank void. And from an evolutionary standpoint, this graceful degradation is just an absolute lifesaver. It essentially boils down to a life or death choice. Would you rather have no photo or a blurry photo? Imagine you're an early human trying to survive in the wild, and you run into a predator. It is vastly, immeasurably better to have a low-resolution, slightly blurry memory of a dangerous animal and exactly where its cave is, than to fall off a memory cliff, have your mind wiped totally blank, and forget the danger even existed. By repurposing our spatial GPS grid into an abstract memory clothesline, the human brain guarantees that a catastrophic wipeout literally never happens. Which brings us to a seriously exciting, paradigm-shifting frontier.
If the deep, fundamental secret to human memory relies on this rigid, completely separate mathematical scaffold, this sturdy clothesline built right out of our spatial navigation systems, could mimicking the hippocampus be the ultimate key to fixing the next generation of AI? As our AI models get larger and more complex, maybe the answer isn't just frantically pushing more raw data into them. Maybe they actually need a biologically inspired grid to securely hang all that data on. It's a wildly thrilling prospect. Well, I hope this explainer gave you a really clear, fun look at the incredibly elegant ways our brains protect our memories. Keep exploring, keep learning, and I'll catch you on the next one.